My dear students, welcome to my today's talk on influence of yolk on the cleavage pattern. I am Dr. MEERA B.K, Associate Professor and Head, Department of Zoology, Maharani Science College for Women, Palace Road, Bengaluru. My cell phone number is there on this slide, so also email ID. After the class, if you have any queries to be sought, do contact me over my cell phone or through my email. Now, let us start the class. Today's topic is effect of yolk on the cleavage pattern. But before getting into the topic as such, we need to know the classification of eggs based on the amount and distribution of yolk as background information. Here, all of you by now know that egg or ovum is a female gamete and yolk is a stored food material present inside the yolk, inside the egg, which meets the energy requirements when the young one develops. Biochemically, yolk is a lipoproteinaceous material. Now we should know how the animal eggs are classified based on the amount of yolk. There are 5 types of eggs which have been classified based on the amount of yolk. First one is alecithal. A means no, lecithin means yolk. Here, yolk is completely absent and such an egg practically do not exist at all. The second type is microlecithal or oligolecithal or meolecithal egg. Micro means small, little, oligo means few. So these eggs are characterized by very little or scanty yolk. All echinoderm eggs, tunicate eggs belong to this type. Third category is mesolecithal or mediolecithal egg. Meso means moderate, where the yolk is present in moderate amounts and all the amphibian eggs like frogs, etc. belong to this category. Then the fourth type is macrolecithal or megalecithal or polylecithal eggs. Macro means large. These eggs are characterized by large quantities of yolk. Eggs of reptiles, birds and monotremes mammals belong to this category. And we have one more type, secondarily microlecithal eggs, also characterized by very little quantity of yolk being present in the cytoplasm. Eggs of eutherian mammals belong to this category. Why they are called secondary? Because when there is a shift from oviparity to viviparity, that is from egg laying phenomenon to giving birth to the young ones directly, there developed the concept of placenta to take care of the young ones. As a result, the eggs became secondarily microlecithal as much reserved food material is not needed for the development of the young one. Hence, the eggs of eutherian mammals are secondarily microlecithal. Then, based on the distribution of yolk, 4 types of eggs have been classified. One is isolecithal or homolecithal. Iso means same or equal. Homo means uniform. Here, whatever little quantity of yolk is present, it is more or less uniformly distributed throughout the cytoplasm. And all microlecithal eggs invariably are isolecithal eggs. Then the second type is centrolecithal, where yolk, whatever is present, is concentrated in the central region of the egg. Insect and other arthropod eggs belong to this category, especially the eggs of silkworm, Bombyx mori. The third type is telolecithal or moderately telolecithal eggs. Telo means tail, where whatever yolk is present, it is present more towards the vegetal pole. It accumulates towards the vegetal pole due to the density gradient. All mesolecithal eggs are invariably telolecithal eggs. Then the fourth category is heavily telolecithal, where the yolk occupies almost all region of the egg and the yolk-free cytoplasm is confined to a small area in the animal hemisphere. All megalecithal eggs are invariably heavily telolecithal eggs. I have presented here in this slide the pictures of different types of eggs which have been classified based on the amount and distribution of yolk being present. Figure A depicts the microlecithal and isolecithal egg. Figure B depicts mesolecithal and telolecithal egg, typically frog's egg. Figure C tells you how a polylecithal and centrolecithal egg looks like, insect egg. And figure D shows how a macrolecithal and telolecithal egg looks like, that is reptilian egg or a bird egg or the eggs of egg-laying mammals. Now with this background, let us study the influence of yolk on the cleavage pattern. Now I have already told you what is yolk, and you should know what is cleavage before getting into the study of the impact. Cleavage is the division of zygote by a series of mitotic cell divisions into a multitude of cells which become building units of future organism. And this cleavage is called by various names like cellulation or segmentation. And here I should tell you what is a zygote. Zygote is a diploid cell formed by the union of haploid sperm and the haploid ovum during the process of fertilization. And this zygote undergoes the developmental processes, and the first stage in the developmental process is the cleavage. Now we should know how yolk interferes with the cleavage process. The yolk granules or globules do not participate in cleavage process as such, but has a profound deterring effect on the cleavage. See, yolk is an inert, non-living lipoproteinaceous material normally present in the form of either granules or in the form of globules. Now, when the amount of stored yolk increases, the amount of active cytoplasm in an egg decreases. The position of the nucleus is variously affected. The nucleus will shift to eccentric position from its central position with the presence of large quantities of yolk in the egg. Then the mitotic divisions of such a displaced nucleus results in unequal cytokinesis, and therefore there is formation of smaller yolk-free micromeres and larger yolk-laden macromeres. Both micromeres and macromeres are the blastomeres. The daughter cells resulting out of cleavage are called blastomeres. They are micromeres if they are smaller and macromeres if the size is bigger. The yolk granules or globules, behave entirely passively during cleavage, and therefore, when yolk is abundant, it tends to retard the cleavage process. The blastomeres rich in yolk tend to divide at a lower rate and consequently remain larger than those which have lesser yolk. The yolk in an uncleaved egg is more concentrated towards the vegetal pole, that is lower hemisphere of the egg, in contrast to animal pole, which is the upper hemisphere of the egg. And this settling down of the yolk more towards vegetal pole is due to density gradient. At the vegetal pole, cleavage is most retarded where the blastomeres are of larger size. Thus, it is needless to say that the amount and distribution of the yolk decides the cleavage pattern, and accordingly, the following types of cleavages have been identified. First one, total or holoblastic or complete cleavage, where the cleavage furrow bisects the entire egg, and this total cleavage is further divided into two types. Equal holoblastic cleavage, which normally occurs in micro and isolecithal eggs. It produces blastomeres of equal or approximately equal size, and this type of cleavage is seen in Amphioxus, which is a protochordate, then marsupials and placental mammals. Marsupials means egg-laying mammals like echidna and platypus. Not egg-laying mammals. Marsupials are another group of mammals where you find equal holoblastic cleavage. Then unequal holoblastic cleavage, the second type. It is seen in meso and telolecithal eggs. The holoblastic cleavage produces unequal sized blastomeres. The smaller blastomeres, which are yolk free, are called micromeres, and few large yolk laden blastomeres, which are formed towards vegetal pole, are called macromeres. This is seen in case of lower fishes and amphibians. Then the second type of cleavage is partial or meroplastic or incomplete cleavage, in contrast to total or holoblastic or complete cleavage. This is seen in macro and centrolecithal eggs. Here, the cleavage furrows divide the small amount of active cytoplasm of the animal pole or periphery of the egg. Most of the yolky portion of the vegetal pole or central area of the egg remains undivided. There are two types of meroblastic cleavage. One is discoidal cleavage, which is seen in macrolecithal and heavily telolecithal eggs. The cleavage remains restricted to the small disc-shaped active cytoplasm of the animal pole. This is seen in elasmobranch fishes, bony fishes, reptiles, birds and monotreme mammals like echidna and platypus. Then another type of meroblastic cleavage, which is exemplified by insect eggs, is the superficial cleavage, which is seen in centrolecithal eggs, where the cleavage remains restricted to the peripheral cytoplasmic region of the egg. Then, after studying different types of cleavage patterns, due to the amount and distribution of yolk in the egg, scientists came out with certain laws which are studied under laws of cleavage. Certain laws have been formulated to highlight the impact of yolk on cleavage. One such law is Hertwig's law, formulated by O. Hertwig in 1881, which states that the nucleus and achromatic figure, that is mitotic spindle, occupy the center of protoplasmic density of the egg or blastomere in which it lies. Thus, in micro and isolecithal eggs, the spindle is located centrally, whereas in a telolecithal egg, it is more nearer to the animal pole. A corollary has been given for this Hertwig's law to make the facts clear. The corollary states that blastomeres divide into two equal parts unless the yolk is unevenly distributed in them. Then the second law is known as Balfour's law, formulated by the scientist Balfour in the year 1885. And the law says the speed or rate of cleavage in any region of the egg is inversely proportional to the amount of yolk or deutoplasm it contains. The cytoplasm containing the yolk is called deutoplasm. In telolecithal eggs, blastomeres at the animal pole divide faster than those near the vegetal pole because they are yolk free in animal pole, whereas they are yolk laden in the vegetal pole. Now, after having studied all this, I want to give a comparative account highlighting the impact of yolk on cleavage pattern. Firstly, we should remember that the cleavage is confined to active protoplasm of the cell. Secondly, the yolk is non-living and inert and has no role to play during the process of cleavage. Bradley M. Paton, a famous embryologist who has contributed lots to the studies of chick embryology, has rightly commented that the amount and distribution of yolk will determine the type of cleavage. Then, in the eggs of Amphioxus, let this be the first case what we study, the amount of yolk is very little and is slightly concentrated towards the vegetal pole, microlecithal and isolecithal egg. So, the yolk does not interfere in cleavage process at all. The resulting blastomeres will be of equal size, hence we call this cleavage as equal holoblastic cleavage. The second case, the eggs of frogs and other amphibians contain moderate amount of yolk which is concentrated towards vegetal pole. Therefore, cytokinesis is considerably modified. Cytokinesis is division of cytoplasm. Karyokinesis is division of nucleus. At the animal pole, cleavage proceeds at a faster rate due to the yolk-free cytoplasm than the vegetal pole, which is crowded with yolk. This automatically results in unequal holoblastic cleavage. The size of blastomeres is very pronounced from the third cleavage onwards. So in frog, the first and second cleavage is holoblastic equal, whereas from third cleavage onwards it becomes holoblastic unequal. Then the third case, the egg of reptiles and birds, the amount of yolk is so large that the yolk-free cytoplasm along with the nucleus is confined to a small discoidal area in the animal pole, and this region is known as blastodisc. Here, the cleavage shows profound modification and the yolky region of the egg is never divided. Hence, it is meroblastic or incomplete cleavage. The divisions are confined to a small discoidal area, that is blastodisc, lying over the yolk mass. The type of cleavage described here is discoidal meroblastic cleavage. Finally, the fourth case, the eggs of insects. The hemi amount of yolk is concentrated at the center of the ovum and the peripheral cytoplasm is yolk free. Therefore, the cleavage divisions are confined to the surface layer of the egg only, and the central yolky portion of the ovum remains undivided. This type of cleavage is called superficial meroblastic cleavage. Thus, students, today we have learnt the classification of eggs based on the amount and distribution of yolk in the eggs, and then we have studied how this yolk influences the cleavage type and pattern. Thank you for your patient listening. Any questions? Don't hesitate to contact me or my cell phone or email ID. Stay blessed. Thank you.